Good evening. I'm Patricia A, Director of the Lloyd Library and Museum in Cincinnati, Ohio. I'd like to welcome you to tonight's program, Fungi as a Source for Drug Discoveries, with Professor Nicholas Oberlies. The Lloyd Library and Museum was founded more than 140 years ago by the Lloyd brothers, three pharmacists who worked in the field of nature-based medicine. One of them, Curtis Gates Lloyd, was also a mycologist who collected books dating back to the 1600s on fungi, along with specimens at the turn of the 20th century. Today, we continue to support research and education in nature-based medicine. I first met our presenter a few years ago when he served as the president of the American Society of Pharmacognosy. For those of you who the word pharmacognosy is not in your vocabulary yet, it relates to the study of drugs and medicine derived from natural resources. It includes plants and also fungi, which incidentally may have been one of the biggest impacts on the survival rate in the 20th century due to the discovery of penicillin and the antibiotics revolution that followed. As we'll learn tonight, many of those compounds produced in nature's factories are then synthesized. Our speaker, Dr. Nicholas Oberlies, is the Patricia A. Sullivan Distinguished Professor of Chemistry at the University of North Carolina, Greensboro. His lab focuses on the discovery of new drug leads and the analysis of herbal remedies. This includes support from the National Institutes of Health for using fungi as a source for anti-cancer and antimalarial leads, and the analysis and quality control of herbal remedies. Prior to joining UNCG, he spent more than a decade at the Research Triangle Institute, mentored by Drs. Monroe and Mansukh Wani, co-discoverers of the anti-cancer drug Taxol. Dr. Oberlies is an internationally recognized expert in the chemistry of nature and medicines from natural products. He's consulted on the analysis of natural products for clients ranging from Fortune 500 companies to biotechs. He's served as a qualified expert witness and on the scientific advisory boards and several journals, and has participated in over 100 invited scientific presentations, both nationally and internationally, and published more than 220 manuscripts. It's now my great pleasure and honor to welcome Dr. Nicholas Oberlies. Hi, Nick.
Oh, hello. Sorry, I was pushing the unmute button. Hello. Nice to see you all. So it's a great pleasure to give a presentation to all of you out there in the masses on behalf of the Lloyd Library. But one thing I wanted to add about the Lloyd Library is it's a really cool place. If you happen to be in the greater Cincinnati area, go check it out. I wish I was a little bit closer. I would love to take my students there. Okay, I'm going to share my screen and try to talk through these slides. Okay, there we go. So hopefully you're seeing something that says fungi as a source for drug discovery. So this is what I'm going to talk about today. This particular picture is from a manuscript that we published about maybe 2 years ago now that talks about actually doing fungal co-culture. So two fungi fighting it out in one petri dish. So we're going to come to that later. But what I'm going to try to talk about today is I'm going to tell you a little bit about why we study and love fungi. And then I'm going to talk briefly about some tools and methods that we use so that you can understand how we're going about looking for molecules from nature that could be used to help humans. And then basically doing that, showing you some examples of how we actually do some of that work. When you talk to an audience like this online, of course, I can't see any of you, which is quite a bummer, but I wish I could. But it's hard for me to exactly gauge what everybody's expertise is. So if I'm speaking a little bit too sophomorically, just put something in the chat if you want to hear some more details, or if my details are way too two in the weeds, put those in the chat as well. And Patricia will let me know how to sort of gauge my talk. I always like to start off with this slide. These are, she mentioned that I worked at the Research Triangle Institute. I spent over actually almost 11 years there working for Doctors Wall and Wani. And they discovered the two molecules that you see here, which are Taxol and camptothecin. I'm going to talk a little bit more about the impact of molecules from nature, especially from fungi, in a second. These two were actually from a plant. The one in the upper right-hand corner is called Taxol. It's used for ovarian cancer, breast cancer, a few other cancers, particularly breast and ovarian cancer. It's completely changed survival times. And camptothecin, the one on the lower left, is used for colon cancer and a few other cancers. And I probably gave a presentation about this about a year ago, about the discoveries of Wall and Wani. And I could spend 45 minutes talking about how much of an influence they've had, not just on drug discovery, but influence on like the way I think about doing drug discovery research and how I try to impart that to my friends and my colleagues and my students. But I think a couple things to remember is that you never know what you're going to discover, right? And the interesting thing about the story of Taxol and camptothecin is that the molecule that they were working on was the one on the lower left. And Taxol was kind of the side project. So I always try to tell my students, you never know when your side project becomes your main gig, because the thing that they're probably most well known for is that molecule in the upper right, even though it was the side project at the time. The other thing is it took a long time for those things to go from discovery to actually being used in human beings. And you know, sometimes people talk about that with kind of a badge of honor, like, oh, they waited 20 years or 30 years. I guarantee you that neither one of them wanted to wait that long. They would have been quite happy getting them advanced as drugs in 2 or 3 years. But sometimes it takes a while from nature to figure out how to get something advanced. And so a lot of times you'll see in my talk, I'm going to describe things that we're doing to try to speed that up. One of the major things is try to supply something in a timely fashion. We can pretty much solve the structure of a molecule from nature with less than 1 milligram. If you've got like a fingernail and you sort of do a little scoop in some salt, you know, that's about 1 milligram. Right? That's about all we need. But of course, if you're going to do something and test it in animal models and in vitro models and human beings, you eventually need grams to kilograms of material. And so this is something that we're always thinking about. So anyway, thank you to Wall and Wani, not just for the discoveries, but for the inspiration that they've given us. Okay, if you ever see a natural product talk, you're going to see this slide. And I'm not going to belabor it. All I'm going to do is tell you that this is a pie chart that basically tells you roughly what percent of drugs, FDA-approved drugs, come from nature. And so for those of you who live in the United States, if you go to your local CVS or Walgreens and you walk in there, 25% of all the drugs in that pharmacy, both over the counter and behind the counter. And I'm not talking about herbal remedies at all. Forget about the herbal remedies, just things that you would consider drugs, topical antibiotics, aspirin, painkillers, antibiotics, things like that. 25% of them are from nature. And perhaps the more interesting thing is if when you get into the anti-cancer space and the anti-infective space, it's really closer to 65%, right? That means they're either directly from nature, meaning the molecule from nature is the one that becomes the drug, or more likely, the molecule from nature inspires some ideas that somebody else uses to sort of tweak the molecule. Lots of molecules from nature don't have great drug-like properties. You can envision if they have to have good drug-like properties, you have to get them in solution. They have to go like, you know, in case of Taxol, how can you put it inside somebody's vein without it precipitating out a solution? Things like that. So sometimes molecules have to be modified for those reasons. But the original idea came from nature. So that's what this slide is telling you. 65% of all approved anti-cancer and antibiotic drugs from nature. Now, I like to talk about fungi. We're going to spend just a minute on this slide. Now, I realize some of you might not be chemists. Occasionally, I meet somebody on an airplane and they say, oh, what do you do for a living? And I say, I'm a professor. They say, oh, what kind of professor? Professor of chemistry. And then they get suspicious. They say, what kind of chemistry? And I'll say, organic chemistry. And they'll go, oh man, I was going to be a doctor except for organic chemistry. I was going to be a dentist except for organic chemistry. And I always give them the same response, which is, I wasn't your teacher. I can't tell you what happened, but those are organic structures there. Don't get mad at me if you don't like organic structures. This is basically showing you how all those little atoms connect together. The one in the upper left-hand corner, that is penicillin. I was asking our host before the talk started, how many people are online today? And she said it's about 100 people. And you can make a pretty good argument that about 40% of us would not be alive today if it wasn't for the antibiotics and the antibiotics revolution that was created from it. So let's just presume there's only 60 people online today, right? It had that big of an impact, especially if you look at survival rates in like 1923, which is before the antibiotics revolution. The antibiotics revolution basically happens right after World War II to 2023. Survival rates have totally changed, right? Things that you would consider an innocuous infection killed people before. All right, so that's penicillin. You'll see that little medal there. That's the Nobel Prize. Many people like to point out that Alexander Fleming got the Nobel Prize. It's true, he did. He made the original discovery, actually a co-culture discovery. The co-culture of a fungus and a bacterium. He noticed the bacterium was dying because of the fungus. But it was also some chemists, Florey and Chain, working in the middle of World War II, working in the middle of England while it's being bombed, who actually figured out the structure and how to supply penicillin. So there we go. There's our start of our probably the most important fungal metabolite ever discovered. Cephalosporins, also another antibiotic, are from fungi. The one on the upper right is a molecule called griseofulvin. Many of you probably have never heard of griseofulvin. It's actually an antifungal agent, but you can see I put next to there a little blue thing that says EML. That is a designation from the World Health Organization. EML stands for Essential Medicines List. And so if you were going to start a clinic, like if you've ever heard of like Doctors Without Borders, who go to some country and there's some god-awful thing going on and they need to set up a clinic and they need a list of what medicine should we bring in the clinic, griseofulvin is one of those molecules. We're also going to come back to griseofulvin later because we use it in some of our experiments. Cyclosporin, that's used for organ transplants to prevent rejection of your organ. And then the one in the middle on the bottom, the statins. The statins were the #1 selling drugs in the history of humankind for a long period of time. The history of humankind. Some of my friends, I live in North Carolina. Barbecue is big in North Carolina. There's a lot of pigs in North Carolina. A lot of people eat their bacon in North Carolina. Some of my friends will say, oh yeah, I eat my bacon, but I take my statins. I always tell them I don't know if that's good advice, but of course I'm not a medical doctor. But anyway, if you've got any sort of cholesterol issues, you're probably taking the statins, an idea that originally came from fungi. And then on the far right, on the bottom, is a molecule called fingolimod. That is a molecule that is the first orally active molecule to treat MS. Orally active means you can take it as a pill instead of other treatments for MS are like an injection in your stomach, right? Doesn't sound very fun to me. So in 2010, that was approved as a drug and it was hailed as one of the top 10 medical breakthroughs of 2010. Okay, so there's just an example of some molecules that come from nature, specifically come from fungi. Now, why do we study them? You notice none of those are anti-cancer agents. There really isn't an anti-cancer agent that comes from a fungus that's been FDA approved yet, right? And that's something that we work on every single day. I had a group meeting with my research group this morning. There's about 10 or 15 people in the research group, depending on how you want to count it. They're working every single day to isolate molecules from fungi that I say hope, but it's not hope. I mean, we've got a lot of data that support that they can kill cancer cells. And just because they can kill cancer cells in a little petri dish doesn't mean they can do it in a mouse or a human. So that's always kind of what we're marching towards. So this is what we're working on. And you might say, well, why do you work on fungi? Okay, they've led to drugs before. It turns out they're not particularly well investigated. Roughly, there are 250,000 molecules that have ever been described from nature. That number is probably a little outdated now. It's probably closer to 300,000. Only 5 to 10% of those, 15 to 30,000 molecules, are from fungi. Many, most of them are from plants. Probably the next most would be from bacteria, specifically organisms called actinobacteria. So fungi are pretty underinvestigated when it comes to this. And that's remarkable because this slide is supposed to tell you that there's either 1.5 million or 5.5 million fungi in the world. Mycologists can debate this and fight this and whatever. I always say it doesn't matter because that third bar, the really small one, is the number that have just been given a scientific name. I believe that's roughly 140,000 molecules. Excuse me, fungi that have been given a scientific name. And an even smaller number have been studied for drug leads, right? So they're extremely underinvestigated. To put that into perspective, let's just go with the low end of 1.5 million fungi in the world. You might say, well, how many plants are in the world? There's only about 500,000 plants in the world. Every year, somebody discovers a new plant from some, remote location, and people debate taxonomy of things, but pretty much plants have been discovered. Fungi, everywhere we go, we discover new fungi, everywhere. People often ask me, do you ever go collect in the rainforests? It turns out I don't because I don't have permission to, but if I was able to, I'm sure I would go find new fungi. But it doesn't matter. We collect in all the state parks. I'm going to show you some information about that in a minute. We've discovered not only new genus or new species of fungi, but new families and even a new order of fungi, a new order. If you discovered a new order of a mammal, you would probably get the Nobel Prize, right? But fungi, they're so underinvestigated. Everywhere you look, you discover new fungi. Also, to put that in perspective, they're the second most diverse organisms on the planet. The top most diverse are the insects. Again, people are discovering new insects all the time. So there's somewhere north of 1.5 to 5.5 million insects in the world. Okay, you might say, well, how do you identify these species? Now, one of the things I want to make sure is clear is right here in the middle, you see what's called a petri dish. So that's a fungus growing in the middle, kind of grows out in a radial fashion from the center. Throughout my whole talk, I'm talking about fungi like that. Fungi that we can grow in a petri dish in the laboratory. That doesn't mean I don't like mushrooms. I'm probably going to eat mushrooms with part of my dinner tonight. I'm a big fan of mushrooms. Mushrooms are also fungi. Mushrooms are technically what are called, most of the time, are called basidiomycetes. That thing above ground is the basidiocarp, the thing that you think of as the mushroom. There's also part of it in the ground, which is called the mycelium. We're often looking at that mycelium. The other thing that we're trying to discover are the fungi we're trying to discover are called ascomycetes, and they pretty much grow as a mycelial mass. We do get a fruiting body, but it doesn't look exactly like a mushroom. Okay, so now how do you identify them? Well, we look under the microscope. That's what morphological analysis means. Look at the microscope, and they're based on the shape and the spores, and are the spores smooth, or do they have little ridges on them or things like that? And then we also do something called DNA barcoding, which means we take out some portion of the DNA and we check it against databases to see if it's ever been described before. Again, 90% of fungi are undescribed, so it's a little bit hard to use DNA barcoding, but sometimes it'll get us to the family level or the genus level, right? Kind of gives us, if you will, like a relative. What relation is it to other fungi out there in the world? We can also sequence the entire genome. That's something that we've done as well. And I should show you, this is our picture over here in the lower left. That's Dr. Huzefa Raja, who is a mycologist who works in our research group. And mycologists are great. Mycologists, very rare breeds. There's not a lot of mycologists in the world, but the ones that are out there are very serious scientists and very serious about studying fungi. Okay, we also collaborate. So people ask me, well, how many fungi do you study? Well, we collaborate with this person. This is Dr. Cedric Pierce. He's, excuse me, he has a company called Mycosynthetics. And you can see that he's holding in his hand, kind of at an angle, a test tube. And that test tube is called a slant. If you look really closely, you can see that there's like jello-like material that's kind of at an angle. That's how you store fungi for a long period of time. So that slant that he's holding has one of his fungi. And he has 50,000 fungi that have been collected from around the world. Most of these were collected in the 1980s. And this business has been around for a long time. And basically, their business model is to supply organisms to pharmaceutical companies, to agrochemical companies, so that they can use them as a source of chemical diversity. And can people discover something from them that can lead to a drug lead? One great example is Mycosynthetics discovered a molecule that kills weeds. So it's an herbicide, right? Herbicidal properties. That's kind of a neat thing. That's not something many people study, but they found a very unique molecule from one of their fungi. And it turns out it wasn't only in just one of their fungi, it was in a couple different fungi um, that were probably related to each other that make an herbicide. So one of the things we do is we collaborate with Dr. Pierce, and they have 50,000 fungi. You might ask, how do you store 50,000 fungi? Turns out he stores them at room temperature. You might ask, do they die? Every now and then some percent of them die, but many of them they've had in their library for north of 20, 30, 40 years, right? And they don't just keep one sample, they have redundant samples. And so if one of the samples died, maybe the other one is going to be okay. But there are so many of them to study. There's plenty of us to work on. So we love working with mycosynthetics. The other thing you should know about mycosynthetics is that most of their fungi are from what are called leaf litter. So leaf litter, it's like when you're hiking through the forest, you know how the ground is kind of spongy underneath the trees? That's because leaves and sticks have fallen off the tree, right? It's autumn right now. Leaves are falling off the tree, but those leaves won't still be there 1 year from now, 2 years from now, 3 years from now, 5 years from now, right? Like a piece of granite would be there for thousands of years, but leaves decompose. Well, fungi decompose those things, right? They're called saprobes. They're saprobic. They decompose dead material. And that's basically, if you ask what's fungi's role in the environment, it's probably to recycle carbon, right? Right? The leaves, the tree, the plant sucks in the carbon, puts it into its leaves, grows things, and those leaves drop back on the ground. And the fungi and other organisms decompose it and get those nutrients back in the ground so that the tree can grow some more, right? There's this great cycle of life, right? So most of his fungi are from leaf litter, right? Okay. Another thing that we study are what are called freshwater fungi. So these are fungi from freshwater environments. So you can envision there's the oceans. Those would be marine fungi. There's the terrestrial earth. Those could be soil fungi, or in the case of mycosynthetics, leaf litter fungi. But you might also study fungi from freshwater environments. And this is something that's kind of unique to our research group. It stems from the fact that Dr. Huzefa Raja, this was one of the things he studied in graduate school, and he worked with somebody who was one of the leaders in the world for studying freshwater fungi. And there's a lot of things that we would like to point out about freshwater. For instance, there is not that much fresh water in the world, right? I think it's something like 3% of the world is fresh water. Fresh water also gets things in it like sticks and leaves and things that need to be decomposed, and that's what the fungi are doing. And the fungi that have evolved in these freshwater environments have sort of unique appendages to allow them to live there, right? So things that allow them to float or to kind of swim or to disperse their spores in the water so they can then float downstream. And it kind of depends on the environment, but you can also imagine that we're scientists. We care about things like global climate change. There's not a lot of water in the world, especially fresh water. It's a very finite resource, and they're very underinvestigated. Fungi from these environments are super underinvestigated. So this is where we get them. We've published a review paper. By the way, I gave our host some links, and I'll tell you this manuscript, if you're really interested in freshwater fungi and want to get into it, this manuscript you could download for free anywhere in the world. Okay, and this is just to show you what they look like. You can see that there's a leaf there in the right, and there's special kinds of fungi that decompose leaves. And again, it's autumn right now, so this would be a great time to go collect those fungi. There's also fungi that decompose wood, and we're particularly interested in wood decomposing fungi. And that's what's shown on the left-hand side. So we collect things from these environments. And I think, and you might ask, well, where do people study freshwater fungi? Well, this is a map that we put into that manuscript. And I think it's very telling. First of all, you can see all the spots where people have collected freshwater fungi in the world. And you will see that there aren't that many spots, right? I mean, there's a lot of the world with no spots in it. Okay, so if you happen to be a budding young mycologist or scientist and you want to think about studying something to study, freshwater fungi are super under-investigated. Okay, very, very niche thing to investigate. The other thing that I wish we had kind of done is I wish we had overlaid the home address of mycologists where these collection sites are, because a lot of mycologists will collect in their own backyards. Not literally their backyard, but like, for instance, in North Carolina, we have permission to collect in every state park, right? So you can envision there's state parks all the way at the coast and all the way up in the mountains. And there's a gradient there from sea level up to, probably roughly half a mile or so up in the air, elevation. So there's a big gradient there of different kinds of environments that we could study. So not surprisingly, you'll see a bunch of spots if we zoomed in on this that are from North Carolina. Huzefa is from, he did his PhD work at University of Illinois, Champaign-Urbana. You can see there's some dots there from the Midwest, right? Whole West Coast is barely investigated. South America, no one studied it. Africa, no one studied it. Australia, no one studied it, right? There's a lot of places we could go and collect fungi. So super under-investigated. Okay, now how do we collect fungi? I think this is a really important point. So I want to spend some time on this slide. So there's a picture of Huzefa, by the way, standing in the stream. And he's got like hip waders on. And what you will do is basically walk around the environment and pick up sticks, right? So we're going to pick up sticks, and you can see down there in the lower left, you can see a bunch of sticks there, right? And what we're looking for are the sticks that are already partially decomposed. So if we pick up a stick and we try to bend it, and it bends really easily, that probably means that the stick just fell in the stream, let's say a week ago or a month ago, right? But you know, if you sometimes pick up a stick and it looks really big, but you pick it up and you start to bend it and it flies apart. Well, that's because something is decomposing it, something that has cellulases and lignases, right? And things that can break down woody material. That's fungi, right? So we're walking around the area, we're picking up sticks, we're breaking them. If they break really easily, we collect them. We're kind of, I don't know, superstitious or something about this. I usually break them in half and throw half back. I just want to take half because we don't need very much in the lab. So we're going to leave some fungi there and bring some back to the lab. And we're going to put them in just a Ziploc bag. So you can see we're going to put them in a Ziploc bag. In that bag is going to be like a paper towel that's been moistened so that we can keep it kind of moist. And we're going to seal up that bag and take it back to the lab. We don't need to freeze the sample. We don't have to ship it off right away. Like, you know, it can take 2 or 3 days to get back to the lab. It's okay. People FedEx us things, stuff like that. But basically, we're going to take that woody material back to the lab, and Huzefa is going to clean it up. So the one right in the middle is like a little Tupperware container where he's taken those sticks. You know, they're covered with slime and goop and all kinds of things from the water. He's going to rinse that off with sterile water and put it in a Tupperware container. We're going to put some mothballs in there to keep mites and other things out of it. And we're going to seal that up, right? We're going to put a top on it. And we just let it sit at room temperature. And what he does then is looks at them under the microscope. And so these two things on the right are images of him looking at a piece of wood under the microscope. And let's say he's going to wait a month, and then maybe 3 months, and then maybe 6 months, and keep pulling out the sticks and see what starts to come out of there. And if you look at these two images, you see these like little black spots. Well, those little black spots are the fruiting bodies of the fungus. So the fungus is basically putting out like a little thing that's going to hold the spores. The spores are like the seeds of the fungus. And eventually in an aquatic environment, when it's ready, it would sort of like let those go. And sometimes they're going to float away. Sometimes it's going to shoot it away, whatever it's going to do so they can go find another place to propagate the cycle of life, right? Okay. And then eventually we're going to try to get these in what are called exenic cultures. Exenic is just a fancy way to say a pure culture, right? So if you look at those pictures on the very bottom, those are Petri dishes with different fungi in them. We just code ours sequentially. So G stands for Greensboro. 427 is the 427th fungus that Huzefa isolated, right? And it's going to be sitting there growing in a Petri dish. And that's how we collect them. Okay. Now, if you really look at them under a more higher magnification, you can start to see the spores. And these are just examples of some freshwater aquatic species. I'm trying to show you that they've got appendages and things that help them survive in an aquatic environment. That one on the lower right, you can see that those two sort of like kidney-shaped things, those are spores. And you can almost see like a halo around them. Well, that's like gelatinous material, right? Kind of sticky. So that spore can float down the stream until it gets to another stick and can stick on. And then it's going to start to decompose the stick. Fungi are eukaryotic, just like human beings are eukaryotic, right? It means we've got like a cell nucleus and things like that. So we're actually quite similar to fungi. However, when we eat something, right, we swallow it and it gets into our stomach. Well, fungi don't have a stomach. they secrete enzymes on the outside. And as they secrete those enzymes, they start to decompose that piece of wood and then suck in the nutrients so that it can start to grow and divide and grow and divide, right? So that little fungus has got to float downstream. The spore is going to stick onto a piece of wood and then do its thing to decompose it. So there's also a database. If you really want to get into the weeds of ascomycetes, Josefa keeps up a database of all the freshwater species that have been described in the world, but it's not very many, and it's nowhere close to 1.5 million. Okay, that is the basics of collecting fungi. Work with somebody who's got 50,000, as we do with mycosynthetics, or go build up your own library, as we do in our own laboratory. We do a little bit of both. We have about 1,000 fungi down in the lab here. And of course, mycosynthetics has got about 50,000. And we have different types of fungi. So we're looking in different sort of environmental niches for bioactive compounds. Okay, so now what I'm trying to show you is kind of 2 chapters to the book here. On the left-hand side is how we extract things. On the right-hand side is how we isolate things and characterize them. I'm not going to get way into the weeds on this, but again, I want to stress, we're not collecting the fungi from the wild and then taking them to the lab and doing all the chemistry. Instead, we're collecting them from the wild, isolating them, getting them to grow in culture, right? And this thing that looks like these Erlenmeyer flasks, let's see. Okay, you should be able to see a little pointer now. These things that look like these are called Erlenmeyer flasks. Those are fungi growing in a flask. One of the things that we learned from working with mycosynthetics for a long period of time is that we grow our fungi on solid substrates. So we grow them on things like rice or oatmeal or breakfast cereal like Cheerios. We take rice, we autoclave it. Autoclave it just means we sterilize it, so it's like cooked rice. And we take the fungi and we sort of pour them in a liquid state on top of that rice. And once again, we're forcing the fungus to decompose that rice for its food source. By doing so, we think that enhances the ability for the fungus to make what are called secondary metabolites. And secondary metabolites are what you would call drug leads. All those little structures I showed you, those are secondary metabolites. Then we do some things like we partition them between solvents, and then we test them against biological assays, right? So this upper right-hand panel here, you can see all these little vials, right? So we take an extract of a fungus. An extract of a fungus is usually brown and gooky. Gooky is a technical scientific term, which means there's a whole bunch of stuff in there, right? And we try to purify it. And we have different things that we do to try to purify it. And as we're purifying things, we will send them off for biological activity. Because when we purify it, there's nothing like the little molecule doesn't raise its hand and say, hey, I'm here, isolate me, right? Instead, what it does is we test it against cancer cells, or we do that with some collaborators at the University of Illinois at Chicago, or collaborators at Ohio State University, or maybe we test it for malaria, which we do with some collaborators at the University of Georgia. And basically, we're looking for a yes-no response. Either yes, the cancer cells are dead. That's good to us. Or nope, they lived. Yes, the parasite that's the cause of malaria is dead. Or nope, it lived, right? And really, when you test things, 90% of the time, they're not active. They do not kill the cancer cell. They do not kill the malarium. They do not kill the bacterium. But every now and then you find a sample that's like the little bit of gold there, this thing that's shining right in the middle. The one that is doing something interesting, killing a cancer cell, let's say. Well, then we're going to do a whole bunch of things. I'm not getting into the weeds of this, but if you want to know some fancy abbreviations, NMR, nuclear magnetic resonance, mass spectrometry, MS, all kinds of things till we can eventually get down to a structure. And that's what we're doing every single day in the lab. So we've got some people doing the mycology, isolating fungi from the environment, growing them on a larger scale. And we've got some people doing the chromatography, the purification, and all of this characterization work so that we could eventually draw the structure of a molecule. We've done this a lot for the past 15 or 20 years. We've gone through more than 5,000 fungi. I think the number is about 7,500. We've isolated more than 500 compounds from fungi. I think it's about 700. Of that 700, about a third are new to science. But even the 2/3 that aren't new to science, it doesn't mean you can find them in a catalog somewhere. Most of them you cannot find anywhere. Somebody published a paper on it in 1974, but that was it. They're retired now. Nobody's got that molecule. And that's the great thing about science in 2023 is that there's all these new biological assays, all this new technology that's been developed that you can now test things against. So sometimes we're taking what you might consider an old molecule and now testing it in modern bioassays. And I would say that we have 5 leads that we're working on. When I say 5 leads, let's go back to Doctors Wall and Wani. At some point, Dr. Wall and Wani, when I knew them, they were my superheroes. They were heroes to me. They discovered molecules that became drugs. But at some point, they had just discovered molecules which had not become drugs. Okay, that's where we are right now with these 5 leads. But what it does mean is that means somebody in the laboratory is thinking every day about how can we make more of that molecule? How can we derivatize it? Remember, I told you that a lot of natural products don't dissolve in something that you would want to put inside your veins, right? So how can we make it dissolve better? How can we make it have a longer half-life so you don't have to take as much of it, so it's circulating through your body for a longer period of time? Yeah. How can we deliver it in a targeted fashion to a cancer cell instead of like, if anybody's ever been through cancer chemotherapy or had a loved one go through cancer chemotherapy, it's not fun. You lose your hair, you get sick, all kinds of terrible things. What can we do to target the molecule so those side effects are not as prominent? So those are things we're thinking about all the time. All right, I'm going to show you a couple of technology things because I've got some videos that I want to show off. Want to see what you think about them. But there's some neat things that we do to analyze fungi. Particularly, we want to analyze the chemistry of fungus right there in the Petri dish. One thing we do is we've got a whole database. We take all this database of all these metabolites, and we've got ways to analyze them really quickly. I don't want to get into the weeds of that, but it's called metabolite characterization. Some people call it dereplication. It was developed by these 2 students on the left who are now both graduated. And today it's run by this guy on the right here. This is Tyler Graff, who's worked in our group for over 20 years. And if you ever need anything fixed, he's the guy who knows how to fix everything and knows how to go through an idea sort of outside the box. Outside of the box ideas is something we want in science all the time. So this is how we do it. We have a whole bunch of data. This is just to show you some examples of molecules and the data that we collect. Again, I don't want to get into the weeds of it, but there's many, many different molecules that we've isolated. Some of them have a molecular weight of around 300, which would be kind of small for a natural product. Some of them have a molecular weight around 600 or 700, kind of medium size. Some of them have a molecular weight around 1800, kind of big. But we're always analyzing these, trying to build up this database. One of the things we do is we try to study them right in. So we take all that data I was just showing you, right? All of that characterization data, and we want to study it right in the Petri dish. So this is a technique that we adopted from some collaborators at the Oak Ridge National Laboratory. You might have heard of Oak Ridge National Laboratory. A lot of early research on nuclear weapons was done during World War II at Oak Ridge National Lab. Today, these guys work on all kinds of things, and they develop something called the droplet probe. And I want to show how it works. It basically takes a syringe, and that syringe will have like 4 or 5 microliters. It's a very small amount of solvent in it. It's a mixture of methanol and water, and it's going to sort of analyze it over your Petri dish. So we're going to sit there and make a little concentrated drop of it. And then we're going to take that little droplet and inject it into an instrument called a UPLC, and it's going to go through a column, and then we're going to analyze it by mass spectrometry and UV. And again, I don't want to get into the details, but you can imagine we're collecting a lot of data just from that Petri dish. We didn't do any extractions and isolation yet. We just took the Petri dish right into the laboratory and analyzed its chemistry. I just want to show you how it works because it's kind of neat, and it could also give you the image of what a fungus looks like. If I was going to say the big difference between studying bacteria and fungi, bacteria kind of grow in a uniform fashion. Not always, okay, there's exceptions, but they kind of grow in a uniform fashion. When you look at this fungus, you're going to see that there's almost 3-dimensional topography, little blips and little places where it's growing up, different kinds of things. Well, if you see different things with your eye's pretty good at noticing things. Those different kinds of things might have different kinds of chemistry, right? So that's something we can do with this droplet probe. So let me show you how it works. All it's doing right now is getting some solvent in the needle, and it's going to go over on top of this Petri dish. So that's a Petri dish of a fungus there. Okay, that's a student. She graduated recently. She now just works up the street from the Lloyd Library at Procter & Gamble. And you can see that she's very carefully getting that needle right above the fungus, We had to build that joystick in the laboratory. That's a fun thing to justify. Why are you buying a joystick? Okay, now this is sped up a little quicker than it is in real life, but probably goes 3 times. This is 3 times faster. But you can see that droplet's going in and out, and it's making like a tiny little extract. And then it's going to go over to that little silver port, and it's going to be injected. And it's going to have that long tube coming out of the right. That tube comes all the way over to that stack of instruments there. And now she's going to analyze all kinds of chemistry. All right, we love the droplet probe. We love, love, love it. We can use it to analyze things in a targeted fashion, meaning we know the molecule we're looking for. So if we go back to that example of mycosynthetics, I told you they have 50,000 fungi. They don't know the identity of every fungus, right? And sometimes the fungi are redundant. So they might have many fungi, many different fungi from different collections around the world that make the same molecule. To us, I think of it like humans. All humans are Homo sapiens, right? Now, if you looked at me, I'm just an average size Homo sapien. I'm about 5'10". I love basketball. I grew up in Indiana. I really love basketball, but I promise you, I can't dunk. In fact, I'm not even very good at basketball. But I work on a college campus. There's guys on this college campus who are 6'11". They can dunk, they can shoot, they can swat the ball, they can do all kinds of things. Genetically, that guy is a Homo sapien and I'm a Homo sapien, right? But he's got the genetic material to be a little bit taller and to be great at basketball. So sometimes we are looking for fungi that have got the genetic code to make a molecule better than other ones. That's a great redundancy. We went, for instance, with mycosynthetics, we went through 50 different fungi, different strains of fungi. They might have even been the same species, right? But different collection points that all made the same molecule. And about 3 of them made it really, really, really well. So that's one thing that we're doing. The other way we use this droplet probe is in an untargeted fashion, meaning it's agnostic. Like we don't really know what we're looking for, but we're looking for something interesting. The fungus is doing something kind of weird, and we want to see, can we ascribe the chemistry to that? Okay, so we love the droplet probe. If you really wanted to get into the weeds of the droplet probe, we've written a review paper about it. You could download that review paper if you wanted to. All right, I'm going to show you kind of a cool experiment we did. Remember me telling you about Griseofolvin? Griseofolvin is this molecule up here that's got antifungal properties, right? It's actually fungi static, so it slows down the growth of other fungi. We isolated it from this tree. This tree is called pawpaw. Pawpaw grows in Indiana and North Carolina and Kentucky, Ohio. Maybe you've seen the fruits of it before. We isolated the fungi from this tree. So that's a type of fungus I didn't tell you about, which are called endophytic fungi, endosymbionts, fungi that live inside the plant, but don't cause the plant harm. They're not killing the plant. They're just hanging out. Maybe they're helping the plant. Maybe they're just waiting for the plant to drop its leaves and then it's going to decompose those leaves. We don't really know what they're there for, but they kind of do some cool chemistry. So we had a fungus that made Griseofolvin, which has got antifungal properties. And I think this is a cool thing that shows you what fungi do. So fungi grow from the middle out. So the oldest part of the fungus is right here in the middle where this black dot is. And so we took that little droplet probe and we studied the chemistry horizontally, and vertically, right? And we color-coded the spots based on the concentration of Griseofolvin. So the youngest part of the fungus is out here at the edge. And you can see at the youngest part of the fungus, it's got the most red. So what that means is that the fungus is sort of pushing the Griseofolvin out to the edges. I recently traveled to Brazil. I was giving a talk down there at a conference, and I had to get through an airport really quickly, right? You ever had to go through an airport really quickly, and you know how you've got to like weave through all these people, and you want to, you don't want to knock them over, but you might want to politely push them out of your way, okay? Well, that's what this fungus is doing here. And in nature, I think this is an important point. People always think, oh, natural products are natural and safe and whatnot. Well, okay, they're also can be toxic. Fungi, if they're going to land on some little spot and decompose that leaf, they don't want some other fungus to decompose the leaf. That is their food source. So maybe the fungus is making Griseofolvin to push away other organisms so that it can then decompose it for its food source. Okay, really quickly, one of the things that we wanted to do then is to study fungi in co-culture. That's why I had that image of 2 fungi fighting. We had one fungus. I didn't go through the details of this. It's an Aspergillus species. It makes mycotoxins. We had another fungus that made Griseofolvin, and we wanted to know what happens if you put them in co-culture, right? This is just to show you that we can analyze the chemistry. I'm not going to go through that in detail, but one of the things we noticed is we put them in co-culture, we would get this kind of wall. that's developed, right? You see that little wall where it's a lighter color? Now, remember, I told you, we've got this device that can analyze the chemistry of things in situ. And if we let that grow longer, the one on the bottom never overgrew the one on the top, and the one on the top never overgrew the one on the bottom. The wall just got thicker. There's got to be some chemistry going on there, right? So we analyze this, and sure enough, if you look really closely, so we analyze these spots down here that are green. In this case, the color coding is just it's not showing you concentration, it's just to show you the colors to the left. But even if you don't understand chemistry data, what you should see is that where we sampled here, there's a lot more peaks than if we sampled at these other spots. And one of those peaks is griseofulvin, which is the antifungal compound, right? Another thing we did is we sampled the top. Okay, here's the Aspergillus species. This is the chemistry we see. We sampled the bottom, not a lot of chemistry that we see there. When we sampled in the middle, we get all these peaks, and we could only ascribe 2 of them to peaks compounds that we knew of. We have a database of 700 metabolites, 2 fungi that we studied extensively, and only 2 of those peaks we could identify. And if you look really, really closely, there's a bunch of these little stars. Those little stars were things we had no idea what they were. You don't know what something is as a chemist, you want to figure that out. Okay, now I want to show you some time-lapse photography. These were our fungi, but we worked with a collaborator. I'll give you his name in a minute if you want to check him out. He's got a really cool website where he does these cool time-lapse photography. On the left is the Xylaria species. It's the one making griseofulvin. You will notice that it grows slower. The one on the right is the Aspergillus species. It grows faster. See what happens when they get close to each other. This is about 2.5 to 3 weeks sped up. How do you like that? One fungus will not overgrow the other fungus, but a little wall forms between them. And if you notice, the one that grew faster, it seems like it's frustrated. It's got to go around the one that's making griseofulvin. I'm just going to hypothesize that somebody wants to see that again. Let's do it one more time. As they get close, notice how the one on the right starts to try to go around the one on the left. And notice that distinct color change between them. Isn't that cool? Let's look at a close-up of that, just because I want you to see that wall that's being formed between them, because that's where the chemistry is happening. See that white? See that white thing that's developing? Yeah, that's what we're really interested in. Yeah, I really appreciate, again, this was courtesy of a guy named Josh McInnes who worked with us to do this time-lapse photography. But that wall is really, really cool, and that's where the chemistry is happening. And that is another one of our drug leads that we're working on. One of the things that we do, I told you that we've got to supply more of them. That's one of the things I learned from Drs. Wall and Wani. Get as much of it as you can. When you do a co-culture experiment in a Petri dish, it's nice, but you don't have enough that you could provide it to somebody who wants to do a pharmacology assay. This is just to give you a really close-up view of how we work on things. We let these 2 different fungi grow in a little bit of liquid culture. This liquid culture is highly rich in nutrients. It's called yeast extract. Oh, I can't remember what the SD stands for, but anyway, it's a yeast extract. Lots of nutrients in there. We're going to create a lot of mycelial mass. We're going to mix them together and then dump them on top of this oatmeal here, and we're going to let it grow for several weeks. And if you look down the top view, that's not a Petri dish. We're just now looking down the neck of the flask. See all that white powdery stuff? That's like that wall that we were seeing before between the 2 fungi. Now, because the fungi are so mixed up like this, that little wall is happening everywhere. That's just to show you that when we do this kind of scale-up thing, remember those little stars I was showing you before? Well, that little star became this big peak right here, which is a molecule we call weldone. Molecule we're very interested in. I'm not going to get into the weeds of how we do this purification, but I really appreciate the work of this postdoc who did some scale-up work and to figure out how to provide more of the material. And basically, we're getting about 1 milligram per flask. So we can routinely, so we grow 25 flasks on a pretty routine basis. maybe it's going to take 3 or 4 weeks to grow those flasks. It's going to take about a week to process it. So about every month, give or take, we can provide 25 milligrams of material. I have a friend who I collaborate with, and she used to joke that about 10 milligrams of a natural product is like a kilo to everybody else, right? So 25 mgs is like 2.5 kilos. So you can do a lot with 25 mgs. We've scaled this up now. We can produce well over 100 milligrams of Weldone. It's not routine. It's not like easy, but it's something that we can do over and over again, right? And so that we can supply material, figure out how to make analogs and do other things. So this is some of the stuff that we're trying to do with Weldone. I think we could eventually generate gram quantities. We could make analogs, might be something you're interested in. We're going to try to get an X-ray structure of it, and we're going to try to understand its pharmacology. And the cool thing about its pharmacology is the woman that we collaborate with doesn't know how it works. Like, she just can't quite figure it out. And that's cool to them, right? You can envision there's a lot of cancer drugs in the world. And so if your molecule works like an already known cancer drug, then it's pretty obvious. But if it works in a way that no one's predicted before, that could be interesting. So that's something that we're working on to this day. All right. So we love fungi fighting. It's something we work on a lot. This is also a review paper you could check out. Let me tell you, I showed a few pictures of people. There's a lot of people that we work with. We have a lot of collaborations. We have to thank the NIH profusely for supporting us. Sometimes people ask me, what's the hardest part of doing science? Do you get frustrated when things don't go well? No. The hardest part of science is getting the funding, right? I stand by that. And so we work with a great collaboration, a great team of people. There's even more people that I should put on this slide. I just ran out of room. So I also want to say we got a great team of people down in the laboratory. Some of these people still work with us to this day. A lot of these students have already graduated. We were actually doing some outreach at a local elementary school in this photo. You can see that we're sort of hamming it up out here by the principal's office, but one of the things we like to do. And if you're more interested in like the weeds of what we do, there's 2 videos that you could watch. The one on the left would tell you a lot about it's sort of me being interviewed about how do we actually do all that chemistry stuff. So if you really want to know about the chemistry, that's one. The one on the right is a shorter video. It was done by PBS, the local North Carolina version of PBS. And it actually shows us out doing some field work and collecting. And it kind of shows you some glimpses of the lab and interviews some of the students and colleagues that we work with. So anyway, happy to answer some questions. And again, it was a great honor. I want to thank the Lloyd Library for putting this together.
Thank you, Nick. That was fantastic. And I do think you should have been my organic chemistry teacher. I might have taken a different career path. Very, very, very fascinating.
I appreciate that.
Couple of things that occurred to me as you were talking. One, when you were talking about the droplet probe, and you mentioned that there was a great redundancy there, and that really feeds into something that I always say, that redundancy is a good thing. So when somebody says you're doing that, that's just redundant. It's like, no, redundancy is a very good thing. And so I love to see another context where that was true.
Yeah, let me follow up with that, by the way, because like this is something that comes up in grant reviews. People say, oh, they got 50,000 fungi, but they could be redundant. Oh, yeah, they could be redundant, but some of them can dunk the basketball and some of them are as short as I am. So we're looking for those ones that dunk, and it really matters. It's a great thing about collaborating with mycosynthetics.
Yeah. Also, you mentioned the pawpaws, which are well known here in Ohio. And in fact, this is a plug for our exhibition next fall. Our fall exhibition is going to be on pawpaws. So,
all right.
Maybe
Asimina triloba.
Yes. Right. Maybe we can find another excuse to have you come to the Lloyd, either virtually or in person. So we did have one question so far. You mentioned that you were permitted to collect fungi at any state park in North Carolina, and that you were not authorized to do so in the rainforest in Brazil. And that's one of the reasons. So we have a question for Bill about why were you not permitted? And sort of the bigger question might be, how does that whole permission process work?
Bill, that is a great question. We get that question a lot. I was just flying down to Brazil, as I said, and someone said, hey, you going to collect some samples? And I said, absolutely not. And the reason it's absolutely not, like, there are some scientists who've gotten in trouble for doing that kind of thing. So the basic deal is there was something called the Convention on Biodiversity. It's like a treaty that was signed in the 90s, and it was updated with something called the Nagoya Protocol. I don't even know, the early 2000s, I would guess. Interestingly, the United States did not sign on to this treaty. So we are not party to the treaty, okay? But many, many, many countries, like if you were to look at the globe and sort of walk around, the equator, like if you sort of went like this above the equator and walked around the globe, you got Indonesia, you got Ecuador, Colombia, all those kind of countries, the Philippines, whatnot. Many, many, many of those countries. In fact, most countries in the world signed on to the Nagoya Protocol. And basically what that means is you need to get permission from the government, whatever government that is, to study their biodiversity. And we can do that. We've had for instance, I still have a collaboration in the country of Jordan, in the Middle East, where we developed a relationship and got all the permissions that I would need to collect the samples. And basically that meant if I discovered something that become the next multi-billion-dollar drug, I would give some money back to the country of Jordan and the people of Jordan. And I think this was a well-intended treaty, if you will. They didn't want people to go interview some indigenous person from the middle of Brazil and steal their indigenous knowledge, if you will, and then go off and turn it into a drug and become really rich. And then, the person who gave you that information from the indigenous part of from the middle of Brazil gets all their trees cut down and they get nothing for it, right? So that was the well-intentioned thing of it. So you need permission to do these things. You can get permission in Brazil. I was just talking to a colleague down there about that, and for her, it's pretty straightforward. So she's a professor like me, studies things like me. She can get permission to collect in Brazil to collaborate with me, but it would be very hard for her to actually send me the fungus. She would have to like extract it and not send me the genetic material, right? So they want to hold that genetic material in country. Good things that have come out of this. Well, I would say that scientists now probably put more effort into training scientists in countries that like developing countries. For instance, in Jordan, we've trained a whole crop of people who are now professors there. And so we love working with that. But so you need permission. So I have permission to collect in the state parks. I got it from the governor, if you will. If I'm going to go collect like at somebody's house, I say to them, do you mind if I collect some sample at your house? Let me tell you a funny thing, by the way, about collecting in state parks. The way I got this little piece of paper, it's taped into our notebook. It says that I have to get permission from the ranger. So I go to a state park. If you're from North Carolina, like I could go to like Pilot Mountain State Park. Thousands and thousands of people visit there every year. And I go find the ranger. There's usually a head ranger, and I explain who I am and what I'm doing, and that I want to collect a few samples. And he says, well, how much do you want? And I hold up a Ziploc bag, a single Ziploc bag, and say, well, that's about all we need. He says, are you kidding me? I go, no. Where are you going to collect it? Out of that stream right there. They'll say, son, take as much as you want. Take as much pieces of wood as you want. So, we only need a little bit to do our science. So.
Thanks. So Cheryl wants to know, how common is it that fungi have Grisio-fulvin-like properties that slow the growth of other fungi? Does this only occur in freshwater fungi?
No, it occurs with many fungi, and we have lots of fungi that interact with each other and do something to each other when they interact. So not every fungus makes that specific molecule Grisio-fulvin, but they do have molecules that will antagonize other fungi. So sometimes fungi are very happy growing on top of each other, but other times they kind of avoid each other. So that sounds like a hand-wavy answer. I'm giving you a hand-wavy answer because I don't have an exact percentage in mind, but I do want to tell you that it's not always Grisio-fulvin, and it's not even always freshwater fungi. It's something we see with fungi from many different environments.
So we have a couple of comments too. Emily says, great speaker, fascinating. I also wanted to point out to people in the audience, if you didn't catch the link that was in the chat, we will be emailing you the links to the various references that Nick made tonight. So you don't have to worry if you missed those.
I just saw another question in the chat from
sorry.
our friend Josh.
Yeah.
It's okay, I can see it, but it's from Josh. Josh is the guy who did the time-lapse photography. Those of you out there, please clap for him. He did a great job with that. He's got a great website. You ought to check it out. He's asking if our database of 700 metabolites is publicly available. Not exactly. Some of that stuff is tied up with intellectual property. However, we do share some of the data with other people, and we share it on sort of a case-by-case basis. So there are many things that we've shared from that library with other researchers, but it's not one of those things that you could just download freely because it's tied up with other collaborations, if you will.
We have a comment from Eric Tepe, who is a medical botany professor here in Cincinnati. You may know him, I'm not sure. Thanks, Nick. Great material for my medical botany class.
Thank you. Appreciate it. I'm glad to know that you're teaching medical botany, so it's wonderful.
Well, Nick, I would like to thank you so much. This, as I said, was
Wait, really before you thank me, can I say one thing?
Sure.
I wanted to say one thing. Are my slides still up? They're still up, right?
Yeah.
Okay, I'm going backwards in my slides. Hold on a second. There's a really important thing I wanted to say about the Lloyd Library. Where is it? Where is it? Where is it? Okay, this is a good example. There's many examples. Okay. I was telling my students this about the Lloyd Library and all the great things that go on at the Lloyd Library. And one thing a few of my students didn't know, but they know it now, is that the Journal of Natural Products, which is shown right here, which is a paper, a journal that we publish in rather frequently, and I review for all the time. And for instance, Cedric Pierce from Mycosynthetics is an editor of, was originally called Lloydia because it was this deep, 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 long collaboration with the Lloyd Library. So if you're ever on, Jeopardy and the $1,000 question is, what was the original name of the Journal of Natural Products? You could say, what is Lloydia? So I'm a big fan of the Lloyd Library and Lloydia, the original name of their journal.
Thank you, Nick. And for those that might be even further interested in that, we have a collaboration that we're working with the American Society of Pharmacognosy to get at least the early couple decades of Lloydia and the Journal of Natural Products available publicly online. So that's a project we're working on right now. So thanks for the shout out on that magazine.
My pleasure.
Well, Nick, thank you again. And I'd like to thank the audience for coming tonight. I'd like to mention that programs like these are supported in part by members of the Lloyd Library. And if you are a member, thank you. If you're not, I encourage you to join, and you can find membership information on our website, lloydlibrary.org. We hope to see you next time. Thanks and have a great night.
Good night.